think everybody's discombobulated from the road work. I'm going to assume that we can all agree on one thing about the moral status of the embryo, and that is that in the space of time from 20 after 3 until 5 or 5:15, we cannot all come to agree on the moral status of the embryo. Nor could we possibly agree on the correct relationship between the answer to the question of the moral status of the embryo and the appropriate legal response of the U.S. government within the context of the particularities of the U.S. Constitution. So rather than talk about that topic in particular, what I'd like to do is have us start this part of the afternoon, at least my presentation, with the assumption that we don't agree about the moral status of the embryo or how the government ought to react. And given that assumption, what can we observe about what has happened and what is likely to happen in the future in the area of embryonic stem cell research? It's worth noting first that historically, this debate, which is closely tied, of course, to the debate around abortion, is one that has dogged the field of embryo research in the U.S. since the 1970s, when we first put into place rather rigorous rules governing research with human beings and put into place additional protections for research on human embryos. And through a series of regulatory actions and inactions during the 1980s under President Reagan and then later under President George H.W. Bush, we had a federal moratorium on any spending that would involve research that destroys an embryo, not for the sake of some therapeutic intent to the embryo, but for the sake of developing knowledge about basic human development or the development of cures for cancer, infertility, or anything else that people might have imagined. That situation led to a rather rapid growth of privately funded work in the infertility area. Many of you here may know somebody who used in vitro fertilization, for example, in order to conceive a child. That's a technique that resulted in many, many embryos being developed and used, and many other embryos being developed and left frozen and unused, all of which happened basically without a great deal of government oversight, and certainly without any government funding to trigger that oversight. By 1992, when President Clinton came into office with a different ideological bent, we were now in a situation in which we first could release money for research on embryos, and second, had a pretty substantial supply of them to work on in the form of these frozen embryos at IVF clinics around the United States. But the politics, again, around abortion, around fetal tissue research, around embryo research, was polarizing enough that even under President Clinton, despite a brief window of opportunity for funding by the NIH, funding never actually emerged. And at the point where it almost came into being, the Congress, following the 1994 midterm elections, which featured a substantial turn to the right in the political complexion of the Congress, withdrew the ability of the president to fund in this area by putting into place certain absolute restrictions on the use of any money that would be involved in research that works directly on the embryo itself. Not the embryo byproducts, but the embryo itself. And so for the moment, once again, as of 1995, it looked like federal funding for embryo research was essentially dead. Now, 1997 is when the Dolly experiment is announced on the front page of the New York Times in February. And then 1998, when Jamie Thompson at UW-Madison makes news with the first successful isolation of human embryonic stem cells in a form that made them particularly usable for research, this kind of immortalized cell line that would replicate and stay in this very blank primitive state so that it was a perfect research tool for all the things that Clive and John were talking about earlier this afternoon. And suddenly, interest in federal funding rose again. In fact, it rose to a kind of fevered pitch. And at the end of the Clinton administration, there was a decision on two things that were rather important. The first decision was that we would begin funding to the extent allowed under congressional restrictions. That meant that although we could not fund somebody like Jamie Thompson to actually take an embryo and derive embryonic stem cell lines from it, because that would violate the congressional restrictions, We could nonetheless, under the interpretation of Donna Shalala, who had been previously UW-Madison Chancellor and was now Secretary of HHS, we could use federal money to work on the embryonic stem cell line sitting in Jamie's laboratory, because that's not working on an embryo. Stem cells are not embryos, and therefore there was a distinction here, and we could take advantage of this distinction. And the second important decision taken by the Clinton administration was to write a set of guidelines for how that money could be spent. And it included rules about paying attention to how the cell lines had been made to make sure that whenever you have a cell line that you want to work on with federal money, it came originally from an embryo that was, of course, worked on with private, non-federal money, but was also an embryo that had been donated freely, no payment, no coercion, no undue incentives of any sort, et cetera. There'd be a set of guidelines for what you could do and what you couldn't do. But before any of that could actually be put into action, and certainly before any money could be released, there was a change of administration, and when George W. Bush came into office, he basically put a moratorium on the Clinton policy so that he could revisit it and make his own decisions with his own advisors. And as you well remember, August of 2001 was a time when people thought that stem cell policy was going to be the signature policy decision of the George W. Bush administration. That, of course, changed radically just a little more than a month later. But for a moment there, it looked like this was the center of the political universe for the Bush administration. And the Bush administration came out with what has turned out to be a rather intriguing effort at a compromise here. On the one hand, they did not want in any way to replicate the Clinton policy in toto. It looked too much like one is taking full advantage of the fact that private funding, or at least non-federal funding, is being used to work on frozen embryos, to create new stem cell lines. Now you come in with your federal money to work on the lines. Of course, you haven't actually used federal money to kill an embryo, but boy, it seems like you're acting in a way that takes full advantage of it. And at a purely emotional level, this was disturbing. Second, there was a fear. It turned out that there was no empirical justification for it, but there was a fear that by having federal money flow to research on the embryonic stem cell lines, that you would actually increase the absolute number of embryos that would otherwise die. And this was an argument that had been used in the 80s regarding research with fetal tissue, some of the very same research Clive was talking about in the Parkinson's area. And it turns out that the decision to discard an embryo or have an abortion is momentous enough that it does not appear to be influenced by the fact that the residual tissue might or might not be used in federally funded work somewhere down the line. But that fear of actually promoting the very activity that you deplore certainly was one of the reasons why the Bush administration took the position that it did. So it said that federal money could be used to work on the very limited number of lines that already existed from embryos that had long ago already been destroyed. And in that sense, the use of these lines would in no way seem to encourage further destruction, but that federal money could not be used on any lines created subsequent to August of 2001, because those would be lines that had to be made with embryos that had yet to be destroyed, right? And that's why we suddenly came to find ourselves in an odd position in which we had so-called presidential lines. The initial estimate by the Bush administration was about 63. It's turned out to be closer to 21 or 22 usable stem cell lines. And these presidential lines are all the ones that existed before 2001, very useful for a whole variety of basic science experiments. UW has a stem cell bank that actually allows for more efficient distribution of many of these lines that can be worked on with federal money. But if you wanted to create a newer line, if you wanted to create a line, for example, that came from embryos that had been diagnosed as having a particular genetic characteristic through the genetic diagnostic procedures that are done at certain places in Chicago and at Wayne State in Michigan, for example. If you simply wanted to have lines that came from a wider array of people with a wider array of genetic characteristics, on the chance that different lines with different genetic makeup would actually have different propensities for differentiation in the laboratory, you could not work on these newer lines with your federal grant. And of course, just to emphasize again, you certainly couldn't develop newer lines with your federal grant. And so what happened is that we wound up having to look for other sources of funding for newer lines and for all new derivations. And those sources of funding came from a variety of places. They came from some of the sponsors you've seen referenced on the slides earlier today, such as JDRF, the Juvenile Diabetes Research Foundation, and the Michael J. Fox Foundation. They came from state initiatives like Proposition 71 in California, which was a bond issue by which the voters of California authorized the government to actually issue bonds that could be purchased. It's essentially a loan from the purchasers to the government of California, which would then in turn be used to finance this research through its new agency, the California Institute for Regenerative Medicine. It was financed through state grants, straight state grants out of taxpayer funds in places like Connecticut, Massachusetts, New Jersey, New York, a wide variety of tactics used for individual states as well as private funders to get into the business. Now, one of the things that happens, and we all know this from our own experiences in our own childhoods, is that whoever controls the purse strings controls the rules. I don't know about you, but in my house, I heard more than once, as long as you live in my house, you're going to do it my way, right? This was a very common theme in my father's house. And the federal government and most funders have the same attitude. If you're going to use our money, you're going to do it by our rules. But remember, the federal government, under the Bush administration policy, was now financing a very narrow range of research on a very narrow range of lines. And so it could only impose its rules on a very narrow range of people and circumstances. Instead, you had new rules coming in from JDRF, from Michael J. Fox, from California, from New York, from New Jersey, from Connecticut, every state, every funder. The National Academy of Sciences, and Mark was kind enough to reference it in the introductions earlier, the National Academy of Sciences worked to develop a set of coherent national guidelines. They have no teeth. If you don't follow them, nothing happens to you. You don't lose your money. You don't get thrown in jail. But nonetheless, to come up with something that had a kind of influential power and set out to create a fairly comprehensive set of rules about how one would approach people to donate embryos, what were the circumstances under which you could ask, what information should you give them before you consider their consent informed, from whom do you have to get consent, given that some of these embryos were made not with the sperm and egg of the people who were actually at the infertility clinic, but often with the sperm of an anonymous donor plus the egg of the woman who's married to an infertile man, or vice versa. You've got anonymous egg donors giving eggs, and so sometimes these embryos have genetic parentage lodged in people who cannot be identified and certainly did not give consent for those embryos to be diverted from reproductive uses to research. So issues about who had to give consent, issues about whether or not you could pay people anything for embryos, for eggs, for sperm, especially since in the reproductive world out there, we typically do see in the private sector payments for eggs and sperm. All of these rules that were written by the National Academies came out in guidelines in 2005. But as is our pattern in America, the land of the eccentric and individualistic, everybody wanted to do it just slightly differently. And so California adopted detailed regulations that implemented 95% of the NAS guidelines, but not all of them. Ditto, Wisconsin's now got a committee that's been working along, trying to develop Wisconsin's own internal rules. That is the University of Wisconsin, not the state. And it looks like they're going to adopt maybe 97% of the NAS guidelines, but not all 100%. And the places they differ from NAS are not the same places that California differs from NAS. And where Massachusetts and New York and New Jersey and all the other funding states differ always varies just a little bit, right? And so one of the things that's happened because of the complexity of the funding situation is that we now have a plethora of regulatory regimes which are largely similar but not exactly the same. And in this kind of patchwork of regulation come a wealth of problems. For example, I'm a researcher at the University of Wisconsin. I want to collaborate. You'll notice on all those slides, they always have a long list of acknowledgments, including people at laboratories in other states and even other countries, right? So here I am at the University of Wisconsin, and I want to collaborate with somebody at Harvard and with somebody at Newcastle in the United Kingdom. But it turns out that the rules in Massachusetts and Newcastle are slightly different. So here we have the following situations. There are all these lines out there we would like to work with, but Wisconsin wants to know the circumstances under which each of those lines were derived. From whom did they come? What information were they given? Did anybody get money? Because they want to know whether those lines meet our standards. And maybe they almost meet them, but not quite. So is that good enough? Do we have some respect for the fact that they met the standards in place at the place and time where the lines were made? Do we have that respect? Or do we say, no, we're going to insist on our own standards? So a kind of question of the exportation of one's own kind of moral regulation of research. Or maybe we can decide that there are a few kind of non-negotiable core principles. We're going to insist on informed consent, but the precise content of the information can vary from place to place. Well, now we've got to debate what's core and what's not. And if something isn't core, then by God, why are we insisting on it here too? Why not just limit ourselves to what's core? And so your committee goes back to work again. Now, run this through not Wisconsin, but Wisconsin, Massachusetts, and Newcastle. And then when each of them decides whether or not they're going to let us use these lines, now we all have to find out whether or not we can collaborate with one another. Because all of our committees probably said, yes, you may, but if you make this slight change, you change the consent form this slight way, you change the protocol that small way, and now we're in this endless loop. Those of you that have gone to law school or have kids that have gone to law school may have heard the phrase renvoi, French for, basically this kind of repeating endless loop in which the question of which law applies, so New York says that Massachusetts law applies, and Massachusetts law says New York law applies, and it goes around and around the court system forever. That happens in research ethics as well. And this is enough to make you weep. Because it's bad enough that we can't agree on the fundamentals of whether this research is right or wrong, or whether it should be funded or not. But having gotten to the point where we agree to let everybody just do their own thing, now we're fighting with the fact that we can't collaborate even among people that have decided to go ahead and do it. So this is one of the effects of the politics of abortion and the politics of embryo research bleeding into the policy decisions about funding, having all sorts of unintended effects that are created by this decentralized pattern of regulation. So I would start simply by noting that if we look backwards and a little bit forwards in terms of the funding situation, we see something that's very creaky and very awkward because of its decentralized and diverse nature. Now, looking forward, you can certainly say that things might slightly improve in the near future in terms of getting harmonization, at least within the United States, because, if you look at the presidential candidates that are still standing, John McCain, Hillary Clinton, Barack Obama, all three of them are on the record as supporting an expansion of federal funding beyond the current Bush administration policy. While none of them, as president, could change the congressional restriction on funding derivation work, because it's Congress that has to change its mind about using money to work on embryos themselves. The policy about only allowing federal money to be used for older lines could be changed by the president with a simple word to his or her staff. So, isn't that amazing? We can say his or her staff. Holy cow, that's really cool. So what we're looking at is the possibility of an expansion of federal funding for this field, and with that would come the opportunity, again, for the federal government to say, if you're going to use our money, you're going to do it by our rules. And suddenly, everybody's going to want the federal government's money, because it's always the biggest source of funding, even if it is currently undergoing some constriction, right? It's still the biggest game in town. So that'll certainly help, although it won't help as much as it would have helped if it happened 8 years ago, because by now, you do have on the books rules in New York and New Jersey and Massachusetts and California that can't just go away with a wave. So the federal rules will apply against a backdrop of state rules that also apply, unless those state legislatures quickly enact statutes that either repeal their rules or say that where federal rules are applying, that we will accept those as fully equivalent and morally, acceptable in lieu of our own. But we've still got that little bit of a hurdle to deal with. Nonetheless, we can probably see some more money flowing. The only real political bump in the road one can see with these three candidates and the funding situation is the one that comes from the discoveries of late 2007 concerning these so-called iPS cells, this alternative way of making pluripotent stem cells, not by deriving lines from embryos, but instead by manipulating ordinary cells of the body, by genetically engineering them to act as if they are undifferentiated, and then moving them forward to the point where you can derive cell lines. That has certainly already spurred a number of people to call for, again, an abandonment of funding for embryonic stem cell research, or to maintain the current restrictions, because after all, quote unquote, we don't need it. It's a rehearsal of an argument that's been made for many years. We don't need embryonic stem cell research because we can use adult stem cells. We don't need embryonic stem cells research because we can use stem cells from cord blood or from placenta, from amniotic fluid. And I would simply urge you during the Q&A section to ask very, very specific questions to the two scientists here about the strengths and weaknesses of each of these different cell types and cell sources. Because as best as I can tell, talking to the scientists, there is simply none, not a one, that is a 100% perfect substitute for all the possible areas of application of all the other cell types. And so there really does not seem to be at a technical level a way to imagine right now a clear road to all of the research and clinical applications you would like to explore if you focus only on one or another cell type and kind of arbitrarily cut off research on another. I'm watching the clock because I know we're running late. I do want to talk about two things that move this from laboratory to clinic that will continue to pose obstacles that are partly ethical and partly political. The first has to do with the move from the lab to the testing in an animal model, and we've already seen some examples of that in the slides that were put up. What may not have struck you immediately when you looked at those slides is that some of this work involves merging human material with animal material in ways that could be rather alarming to some people in some circumstances. Now, we already do have lots of examples of animal-human mergers that we have gotten accustomed to, or at least haven't heard a lot about because it didn't alarm the journalists enough to write too much about it. So you may know somebody who had failure in their heart valves that got a heart valve transplant where it was using pig valves instead of human valves. So we know that you can take animal material and put it in a human. It's not ideal. The immunological rejection problems are exacerbated when you go from a different species into human. And there is a legitimate concern from a public health point of view that the close contact of human and non-human material might possibly allow a virus that exists, for example, in the animal population to mutate into a form that would be particularly dangerous for humans because we never would have seen it, our bodies have no resistance to it, and it could sweep through us and cause an epidemic that we can't control. And this problem of so-called xenotransplantation is one that the Food and Drug Administration does look out for in the world of transplants. But there are other kinds of human-animal combinations that we really haven't thought a lot about and that do get people a little bit nervous because they don't know how to evaluate them. For example, if we want to test out some of what Clive is doing, which involves transplanting cells into the brain, and if we have to go through a stage in which you're going to take human material and insert it into the brain of a non-human animal because you want to see whether or not certain aspects of it will work before you move directly into human experiments where you might kill people. We're much happier generally at killing animals than people, although I apologize immediately to the animal rights people in the audience. But generally we are much happier to go ahead and kill animals to save people. So you put human material into the brain of an animal. Does that get you worried? How much human material do you have to put in before you begin to wonder what exactly have we created? How do we know what we've created? I mean, in many ways, this is where the opportunities for collaboration between psychology, philosophy, and biology departments gets really interesting. Because as far as I know, and I'm happy to stand corrected, we do not yet have agreement on the moral status of an embryo, but we also don't have agreement on the nature of consciousness. Nor do we have agreement on how to measure consciousness, although we have lots of things we do measure in terms of self-awareness and cognitive abilities. I don't know that there's total agreement on how to measure what it is that we can't completely define. And if it is really consciousness that we're worried about when you're talking about brain-brain transplants, then the lack of an ability to know what you're worried about, let alone how to measure whether you got to where you're worried, can actually just worry people to begin with. So when Irv Weissman at Stanford University was putting human material into a mouse and was replacing virtually all of the neurological material with human material, people said, oh my God, I mean, he's made a mouse with a human brain. Except, of course, he hadn't. He made a mouse that had human brain cells. It didn't have a human brain. A human brain is more than just its cells. It's its architecture, and it's the functioning of the cells within the architecture, which only goes to point out even more thoroughly how confused we are about the nature of what it is that allows this amazing organ to give rise to this mysterious phenomenon that we call consciousness, let alone ensoulment, which is even harder to define because only a portion of the population believes in it. So, given that, the experiments that we're about to see having to do with the introduction of human into non-human are going to really pose a challenge. We're used to the idea of oversight bodies that help to make sure that the introduction of new techniques into research is modulated, that risks and benefits are analyzed all along the way. But if these groups are as unsure as the scientists themselves as to what they ought to be worried about, if the guidelines are as flimsy as the ones that we wrote through the National Academy of Sciences, without any further detail, it's still a mystery to me, not only how to define consciousness, but how to define how we're going to review the experiments that raise concerns about consciousness. The one thing that we sort of have agreed upon is that there's a point at which our ignorance is so profound that we're really, really worried. And so the introduction of human material at the earliest stages of primate development, so for example, human embryonic stem cells into a monkey embryo, that gets us super worried because now you'd have human material throughout the entire resulting body, brain and body, and it's into a species that's close enough to us in some primates that the architecture is different, but how different and is it different enough? We're not really sure what the effect would be on the resulting organism, and so that's the kind of thing where we've said, let's just draw a bright line and say, let's not do this until we can figure out better how to predict the results. But there's a world of experimentation that goes from what we're not worried about, taking a pig valve and putting it in a heart, or taking a few embryonic stem cells and seeing if they'll graft properly in a sheep liver. There's a world from there where we don't seem to worry up to putting human embryonic stem cells into a primate embryo. And the whole world in between is the problem. But let's assume we figured all that stuff out, and we got past all that, and now we're ready to go to the FDA and say, let us start with our experiments. I want to conclude by just pointing out that the move into humans is also fraught with problems. Because actually, excuse me, I'm so sorry. I did actually forget to just note one other thing about the chimera works. Let me back up just a moment. Because I did tell you it's ethics and politics, and the politics here, I should just note in passing if you want to discuss it, is the politics around the meaning of biological life and the meaning of humans. So I guess in a sense it does circle around again to the moral status of embryos and of all other kinds of human biological life. Because one of the reasons that people get concerned about this merging of non-human and human material is because of a perception that we might be crossing so-called species boundaries, which leads very nicely into a discussion about evolutionary theory and the degree to which species boundaries is a concept that makes much sense at all. But to the extent that people cling to it as if there really are these really clear demarcations among species, there is a sense that humans are somehow special and unique. The very fact of being human is what gives one importance. That in and of itself is something that may ultimately get challenged if we are able to create more of these combined organisms. So the research is feared not only for the effect on the animals, but also its effect on whether we continue to be able to view ourselves as the center of the kind of moral universe, just the way the discoveries that the Earth revolved around the sun and not the sun around the Earth challenged our notions about the place of humanity in the universe. Let me move on very quickly then to the clinical testing. Clive and John both alluded to this. Clinical testing is a tricky business with humans because the very first experiments are very, very dangerous since you really don't know how well your animal work is going to generalize to humans. And so we tend to go very cautiously. And you might think in an ideal world that we would take Clive's little balancing scheme and we would say, okay, which is the disease we want to start with? Let's see, maybe it's Lou Gehrig's disease because it's terminal and it's horrible and there's no other cure, so for these people, even a high degree of risk is worth it because they don't have an alternative. Even that poses a problem, of course, because when you're that desperate, it often becomes almost impossible to make a reasoned decision. And hope will often trump reason in those circumstances. But let's assume that with enough counseling and enough waiting periods and enough, just enough, kibitzing, we can make sure that these people are given a chance to really think this through. Right? So we're going to start with ALS, maybe we'll move on to Parkinson's, maybe then we're going to move on to these other kind of chronic illnesses like diabetes that will eventually likely shorten your life or reduce the quality of your life. But we may not get to something like a spinal cord injury until way down the list, because with spinal cord injury, after what is undoubtedly a horribly difficult initial period of adjustment, people can have very happy and productive lives. So the risk of losing your life from research is a significant risk in light of the fact that the alternative may be to take the current, not quite standard, but still, better explored function-sparing drugs that are often now given right after the acute injury, the acute trauma injury that causes the spinal cord problem. There can also be, and John was pointing this out to me in the break, there can also be a great deal of spontaneous functional improvement, because right after the injury, often it's not clear exactly how much function will be lost until there's an initial period of swelling and then the swelling going, all these other things that go on. So you might not do spinal cord injury first. Right? Nonetheless, as you heard, Geron out in California has been, promising/threatening every year to go forward with its clinical trial for a stem cell transplant that would attempt to salvage or restore spinal cord function fairly soon after a traumatic injury. And we're unable to actually change the order of the experiments because the FDA is not a public health agency. It doesn't get to put out a request for proposals in which it says we want to see an ALS proposal first, and then we want to see Parkinson's, and then we want to see diabetes. Instead, it sits there like the catcher in the baseball game, and it just waits for somebody to throw the ball, and then, like a referee, they call whether it's a strike or a ball. It's either inside or outside the lines in terms of meeting the FDA's regulatory standards, in which there's some benefit that outweighs the risk. But it may not be the best benefit to risk balance, it just has to be barely acceptable, and then they'll let you go forward. So, in the world of clinical trials, we've got a whole series of dilemmas here. We've got therapeutic misconception, false hope, and lots of hype. So you've got patients calling constantly these guys, and even calling non-scientists like me saying, how do I get this therapy? You've got patients that are going all over the world for sham therapies, and you've got patients that are politically agitating for the FDA to move more quickly to approve these trials. And then you've got the problem of getting people enrolled with some degree of real rational consideration of the risks and benefits. In addition, because much of this work is in fact still in the hands of private funders and private companies, you've got to some extent not just the usual kind of intellectual race for glory and for fame, but you've got a real financial race. So you've got companies up in Massachusetts, for example, that have what seems to be a suspicious habit of making dramatic announcements in the newspaper of advances in this field, always in close proximity to a time when their stock prices are depressed and they need to get an additional infusion of capital from investors. So there is a kind of manipulation of the news from companies that need investors, and they certainly are going to be pressuring the FDA to let them move forward with trials, because by God, that's the thing that's really going to bring the money in. That's certainly going to send the stock shooting up, at least for the moment, until the trial results emerge. And so what I'd simply like to conclude by emphasizing is that even if we were to say, let's wave a wand and magically say that the ethical problems that have plagued us in the past have been solved. We all agree now. I'm not sure what we agree on, but we all agree. It doesn't mean that the problems that we've created for ourselves up to now will go away. We will still be left with a patchwork of inconsistent and irritably, awkwardly interacting funding mechanisms that will dog the scientific community for the foreseeable future when it comes to collaboration. We have the looming threat of the general public, you guys, rising up as you realize that there's about to be this raft of research that involves human-animal combinations that are going to have the cover of Time and Newsweek, I guarantee you, scrambling for the academics in the classics departments to give them the names of all those funny-looking Greek mythology creatures that they're going to want to use for the imagery for their articles. And then we're going to have to deal with the politics of illness, in which patients with terminal illnesses are agitating for faster access to therapies, and companies with a financial interest are agitating for faster approvals at the same time that the therapeutic community, the physician community, is trying desperately to keep a realistic view of the possible benefits as well as the risks of the newer interventions as compared to the existing interventions that they still have in their toolkit. Thank you very much, and I'm happy to turn it over to Cynthia.